Assalamualaikum everyone. As you can see back here, this is algal bloom occurred in a freshwater bodies harming many organisms. And this is what we call as FYUP, freshwater harmful algal bloom. Hi, my name is Aimi Alina binti Hussin. I'm happy to present my final year project, which is under supervision of Dr. Farizal Ahmad Kamarudin with the title of Controlling Harmful Freshwater Algal Blooms Through Fungal Flocculation. First, I'm going to explain about the problem and the objectives for this project as the introduction. Freshwater harmful algal bloom has been dominating in a global scale, and unfortunately the occurrence causes damages to lives as well as the economy. And to our despair, what causes FYUP to occur is unpredictable, despite some of the factors have already been acknowledged. Hence, there are lots of methods has been established, physically, chemically, or even biologically, but all seem to be inefficient. Either they are expensive, not environmentally friendly, or harmful as they are untargeted flocculant. So to date, a very suitable flocculant to control FYUP has not been found yet. Hence, the workload of this project in finding the ideal flocculant has been divided into three objectives. The first objective is to screen, isolate, and identify harmful freshwater microalgae from local environment. Second one is to study the growth performances of freshwater microalgae in 2 liter photobioreactor. Last but not least, my objective is to optimize harmful microalgae flocculation process using one factor at a time. Of fact. Next, on to the literature review part. There are 3 significant factors in causing FYUP: the nutrients, especially nitrate and phosphate, the condition of water that causes agitation, and lastly, the light for the growth of microalgae. As mentioned previously, there are 3 methods of flocculation, but in this project, the fungal biological method is the main focus, while chemical screen are used for comparison with the biological method only. So, biological flocculant use is Aspergillus aculatus because of 3 advantages, which are: 1, it is from Aspergillus; 2, since it is widely distributed in local area, thus it is easily isolated. Meanwhile, chemical flocculant are chitosan, aluminum sulfate, and copper chloride. That missing puzzle has put us in a very difficult position to control FYUP in reservoir and public lakes, where the flocculant should be cost-effective for large-scale production and environmentally friendly. So, with all the information have been compiled here, this project aims to find the most ideal flocculant which does not harm anyone but the harmful microalgae itself. The third part of this presentation is the methodology part. So, this is the overall framework. First stage is to achieve objective 1 by screening and isolating of the dominant microalgae species. Next, to determine the harmful dominant species, morphological identification analysis is used. In stage 2, to study the growth performances in 2 liter photobioreactor, increasing the biomass production, analysis of dry algal cell weight is conducted. At the last stage of optimization of harmful microalgae flocculation, the fungal is cultured and undergone dry cell weight analysis. After that, the harmful microalgae is flocked using fungi and chemical in flock char test. In this part 4, results and discussion, I'm going to present the findings of my project in detail by starting the results for the first objective. So, for first objective, it was further divided into 3 parts, which are 1, sampling and screening, 2, isolating the selected dominant harmful microalgae, and lastly, to identify the isolated microalgae by comparing the morphology with algae database. The samples taken were then taken to be screened roughly under light microscope. The freshwater microalgae species present will differ in their shapes, as some are unbranched filamentous and some are colonial forms. However, Enkistrodesmus and Monoraphidium have distinct shape because they look like needle shape and crescent moon shape. And since there were abundant of microalgae population, samples were enriched in 5 different shape class containing BG11 with fixed temperature, light intensity for about 10 days. Overall, we can initially predict that Oscillatoria species will be the dominant species because of this. There are 3 dominant species in this 5 shape class. Shape class 1, labeled as F1, is dominated by Chlorella species, while F5, conquered by Sundesmu, which both are cultivated due to their high lipid content. And the rest of the class is dominated by a microalgae predicted as Oscillatoria species, which mostly known as harmful microalgae. After that, the shape class with dominant Oscillatoria species were diluted using 6 times zero dilution and grown using spread plate technique. The plates show the filamentous string light, which there is possibility it is Oscillatoria species. Next, to further confirm the culture, the plates were observed under light microscope. The observation shows that there is mixed culture of Oscillatoria, but also confirming again, Oscillatoria is the dominant species. So, after the spread plate, the mixed culture was used to isolate Oscillatoria by streaking many times until unialgal culture was obtained. The majority color of olive green of the filamentous also confirming the Oscillatoria is dominant, even though it is contaminated with zooplankton. But overall, it does not affect Oscillatoria in any way. Then, finally, the morphological identification was conducted by comparing with Oscillatoria princeps. It does have similar trichome, the suppression disc for the homogonium, and apical cell which has the narrow hemispherical shape. Therefore, with that being said, objective 1 is successfully achieved. Okay, since we finished with objective 1, now we proceed with results for objective 2. Before growth performances could be studied, the experiment was started with the study of growth profile in aeration and non-aeration condition. The experiment was first conducted to determine the necessity of aeration during the study of cultivation. Overall, it showed that the aeration was better, yielding 0.5 gram per liter. Plus, if you see in the graph, there is shorter lag phase. The short lag phase may due to the optimized condition where it provides the microalgae to adapt easily to the climate change. So, for onward study, Oscillatoria was cultured in arid condition. Okay, now we proceed with first growth performances, which was in different aeration rates. The graph over here shows that the best aeration rates for Oscillatoria is 1.0 liter per minute, which gives better agitation. It is crucial for microalgae to have optimum aeration to allow equal exposure of the cells to nutrient and light. Also, this is very important to avoid sedimentation. Meanwhile, 1.5 liter per minute and 0.5 liter per minute, on the other hand, yielding lower dry cell weight due to the reasons given. So now, we proceed with different light intensities. From the graph, it illustrated that 6,000 lux, the most intense lux, produce 0.67 gram per liter of dried Oscillatoria species cell weight. This is made due to high lipid production, as reported by previous study where high light intensity induced higher lipid content. But different to 4,000 lux and 2,000 lux, the lower cell production may due to self-shading effect to Oscillatoria since the light intensity is low. Hence, the best light for Oscillatoria is 6,000 lux. The last growth performances study is the nutrients ratio composition where nitrate is fixed but different phosphate composition. In the graph, it shows that the optimum ratio is achieved when the microalgae were cultured in nitrate to the phosphate of 40 yielding 0.40 gram per liter dry cell weight. Meanwhile, too much phosphate provided in ratio of N to P 20, there is low biomass. In fact, it is the lowest. But different case for NP60, where low biomass is yielded because of insufficient nutrient. With that, the objective is successfully achieved, as the growth performances of freshwater microalgae in 2 liter photobiota have been studied. Up until now, 2 objectives have been successfully conducted, which now is the time to achieve objective 3. The flow began with the study of growth profile of the bioflocculant, Aspergillus acaulitus. And at last, the comparison of flocculation efficiency between fungal and chemical was done, achieving the final objective in optimizing harmful microalgae population using OFACT. Okay, so growth profile of Aspergillus acaulitus was studied to report fungus originated from local area which crucial for flocculation. The results is presented right here. In general, pH 4 is the best for fungal culture. It produced highest dry cell weight of 8.7 gram per liter at the 4th day, while the lowest dry cell weight obtained is 4.3 gram per liter for pH 2. As opposed to previous study reported, pH 5 and pH 6 are the optimum range for Aspergillus acaulitus. In this study, pH 5 and pH 6 yield lower biomass than pH 4, which may be because of the different adaptation of the fungus. However, pH 2 as the worst is similar to the study because the study reported that only certain fungi can grow in strong acidic conditions, such as Penicillium species. As it can be concluded that pH 4 is best to grow Aspergillus acaulitus for flocculation. So, next, the flocculation of oscillatory species using Aspergillus acaulitus was proceeded, considering the exponential phase and the optimal condition of both species are known. For bioflocculation, only 2 range of fungal concentration at different pH culture were chosen. So, looking at the graph, 20% fungal culture in pH 4 resulted to highest flocculation efficiency, which is 93.14%. This is because when double concentration of mycelium fungus used, more microalgae cells trapped at a faster rate. Also, it increased the collision, so more charge will be neutralized. And there are only 2 dosage because of these reasons. After that, the flocculation using chemicals were carried out to screen the best chemical for comparison to the fungal flocculation. The graph here shows a significant fluctuation of flocculation efficiency resulted from using aluminum sulfate as flocculant, with highest flocculation efficiency is 96%. The harmful of aluminum sulfate, adding more reason of why it is not a good flocculant. Next chemical is chitosan, achieving slightly lower than aluminum sulfate at 550 microliter, which is the highest concentration tested. It achieved 94.12% efficiency. It can be considered as a good flocculant due to the degradable properties, efficiency, and non-toxicity, but it is expensive, making it is not a good flocculant in a large scale. So, the third chemical used is copper chloride, achieving 93.40% flocculation efficiency at 6% concentration. The advantage of this chemical is it is a cheap flocculant to be used in a large scale area, but from my reading, it causes unexpected events which may be harmful to be used in public area. So the unexpected event is oscillatoria can actually tolerate copper. Hence, this microalgae is used for bioremediation to absorb excessive copper. Besides, using copper chloride could also cause shifting effect to other copper-tolerant microalgae. And with that, in general, chemical is either expensive or harmful for flotation in Sembawang Barat Dam. So before we successfully achieve Objective 3, comparison of flotation efficiency between chemical and biological strength. So this is the comparison in terms of efficiency, advantages and disadvantages. Aluminum sulfate has the highest efficiency, but it is unreliable flocculant. Next is hydrosan, an expensive flocculant to be used, yet it is safe to the environment. Third is CuCl, where the flocculation efficiency is almost same as fungal, and it is also a cheap flocculant, but it is too harmful and may cause shifting effect. Lastly, fungal of 30% concentration, achieving 93.1%, flocculation efficiency similar to CuCl and degradable properties similar to hydrosan, but it required extensive study. So in my opinion, these are some improvement could be done on fungal flocculation. So therefore, with that, Objective 3 have been successfully achieved. Now, we have come to the conclusion part. The harmful microalgae has been concluded that dominant species present at Sembawang Barat Dam is Oxydetra species. Second, there are the conditions where Oxydetra work best, including the dry solvent yield. Next, for biological flocculation, 20% dose of fungi at pH 4 can maximize the flocculation efficiency up to 93%. Third, the chemical flocculant achieve higher efficiency than fungal flocculation, but it is very dangerous to be used and expensive. Hence, Aspergillus aculatus provide the best flocculant, though the efficiency is slightly lower. And these are the recommendations for future work, including combining the morphological and 16S rRNA gene sequence analysis for the confirmation of microalgae species identification. And these are the others. In this slide, these are my references that I used. And with that, I would like to conclude my presentation with thank you. Have a nice day and see you soon.